Okay, hi everyone. I hope you can hear me well. Thank you, Barbara, for the very nice introduction. So everybody can hear me?
I guess so.
So, okay, great, good, okay, good. Now I can also see myself. Okay, great. So, so what I thought I would do today is that I will give you a brief description of the projects we're trying to do here specifically for neuroendocrine tumors. So my group started really 2 years ago, so we are really new, but I think it will become really fast obvious why we got involved in this research with neuroendocrine tumors. So let's see if I can get this going. So the first thing I really want to make clear from the beginning is that I'm a research scientist. I'm not a medical doctor. So I will not be discussing or providing any advice with regards to disease management. And I really try to make this presentation non-scientific. So I hope that you will be able to understand it. And if you have any questions, please put them on there. We can discuss at the end. So where are we? So so this is the Sunnybrook Research Institute, and I assume some of the people in this meeting are maybe patients or they have families members that they have neuroendocrine tumors, and maybe they're familiar with the Odette Cancer Center, which is at the entrance of the Sunnybrook Research Institute Health Sciences Center. You can see here. But the first building you see on your right, which is across from the Odette Cancer Center, is the research building, and my group is on the second floor, what we call the S2 wing. And as I mentioned, we opened 2 years ago. So when I joined, what we wanted to do is to establish what we call a translational research program. So what's a translational research program? That's basically the concept is that you take something from bedside, which is really related to a clinical problem, we'll bring it back to the bench, which is the lab, and then we'll try to understand what's going on, and then hopefully bring it back to the clinic and hopefully have an impact in patient care. So why do we want to do that, and what are our goals here? The goals that we have is to be able to detect the disease early and be able to monitor the progression, so to be able to have early detection of neuroendocrine tumors, to develop personalized treatments and identify new therapies or improve existing therapies. Now, one of the buzzwords are probably, you heard a lot about these personalized treatments or precision oncology. And I'm going to try to explain it in very simple terms. So, you know, the old paradigm, we're going to use fruits here. Let's say we have grapes, but the old paradigm was to put, for example, all breast cancers together and call them grapes, and all neuroendocrine tumors together and call them apples. And then in the old days, we used to treat them with the same approach. But now what we know is that, for example, neuroendocrine tumors like apples, they are not all the same. So, for example, if you have, you know, a neuroendocrine tumor that is the type of red delicious versus one that is honey crisp or the Granny Smith, you're going to have to use a different approach to treat these patients and the disease. So really, that's one of the things we're trying to understand, and that's what we call personalized treatments, and one of the things we want to understand in this translational research program. So I'm going to give you an example here. In my group, we use a lot of models, like mouse models, and this is how a pancreatic neuroendocrine tumor will look into the mouse model. So what you see here is a pancreas, and what you see with green and blue, these are the neuroendocrine tumors, pancreatic neuroendocrine tumors, and we use tools to differentiate them. So here you have the type A and the type B. And you know, if you look at them here, they look at the same size, and you don't see any obvious differences. But in fact, like if you look at during metastasis, so which of these two types will be able to go on and move into the liver and form metastasis is only type B. So here the idea is that what we want to understand is how can we differentiate type A and type B. So if the patient has pancreatic neuroendocrine tumor type A, probably we don't have to use an aggressive treatment. Probably we have to just watch and see how the disease will evolve. But if somebody has the pancreatic neuroendocrine type B, then that's, in this case, we're going to have to probably use different treatments and be more aggressive when it comes to the approach in the clinic. So these are some of the questions mechanistic questions we're trying to understand. How do we differentiate the two types, or there are more than two types, and can we find treatments to treat the more aggressive ones? So why we are trying to set up a neuroendocrine translational research program at Sunnybrook? I think a lot of the people in the audience will be really familiar with the Susan Leslie Clinic of Neuroendocrine Tumors. This is the largest probably clinic for neuroendocrine tumors in Canada. It's one of the largest one in the world. And here you see Dr. Simran Singh and Dr. Julie Hallett, which are two of the members in this clinic. The really big advantage for us as researchers is that this clinic sees a very high volume of patients, and what we want to do is to use some of the material and we'll talk about it, from patients, and try to understand the disease. So this is a slide I borrowed from Dr. Simran Singh. Is a slide that he likes to show, and what in this slide, what he emphasizes is that we're always trying to put patients in the middle of care, and you probably, some of you very familiar of the different approaches they use here at Sunnybrook in order to treat patients. What we like to do is to position ourselves here also as part of this scheme, and what we want to do is to connect with the patients, and that will enable us to develop this NET translational research program, which involves developing preclinical models, new treatments, finding tissue biomarkers, and developing liquid biopsies. And we're going to talk for some of this. So, but you know, the most important thing here is from these two arrows, really, it's the one that goes from the patients to our lab, or the NET translational research. And the first question here when I join is how we're going to connect the patients with my group, and that it will have enabled translational research. And the answer to this is what is called biobank. And the biobank, it's a facility that enables the collection, preservation, storage, and supply of biological specimens and data from patients. These are collected following standard operating procedures, and then these materials are made later on available for research. So if we talk about it in general terms, usually what will happen is that patients will obtain an informed consent from patients. When then these patients go to the clinic, let's say if they have a surgery or if they give a blood, some of the specimen will be collected, will go into the biobank, and some of the sample then will come into my group. So now this is a program that is we established last year in February, and since last June it's up and running. So now every patient who comes at Sunnybrook will be approached by our team here, and this is we'll get this leaflet about the biobanking for cancer research, so shown here. So what will happen is that after a patient will give his or her consent to collect sample. If this patient will go for a surgery, then the tumor, once it's excised, it goes into pathology, and then the first thing that happens is that we store the sample, it's called here as FFPE sample, and then it will go for proper diagnosis. So this here I want to emphasize that always a priority is the proper diagnosis and the proper follow-up for the patient. Now, if there is a tissue left over, then the second thing that happens is that we snap freeze some, so that allows us to preserve this sample, and this goes into the biobanking. And then the leftover, it comes as a fresh sample into my group, where we try to establish different models, and I will talk about them today, which we hope to enable us to understand more the different mechanisms and different types of neuroendocrine tumors. Now, the second thing that we collect is plasma, and usually this one for people who undergo surgery, we collect plasma before. And again, this is done when they have a regular blood draw before surgery. So again, we don't change any of the clinical care. And then this sample again, it will go into the pathology. Some of this is used for diagnosis. Some of this is for biome banking, and the rest of it comes to our good for liquid biopsies. So as I mentioned, this is group, this biome banking, which is really the most important, I think, core for our program. It started 6 months ago. And the Dr. Julie Hallett and Dr. Canby Low are the two surgical oncologists who help us to collect the samples. Here you see Dr. Nilakshi Kulathunga, which is a postdoc in my group and really leads this project. And as you've seen, this is from May to August. We were able to enroll to identify 20 patients who are eligible for biome banking. We enrolled 16 of them. I think today the new numbers are 30 and 20, and we have around 16 tissues. So neuroendocrine tumors, it's a rare disease, but the volume that we have here at Sunnybrook, it really helps us to collect a good satisfactory number of samples, which will enable us to ask the questions we want. So I'm going to start with liquid biopsies to introduce them and tell you a little bit of the things that we are doing here. So what's a liquid biopsy? I think maybe some people here are familiar with traditional tissue biopsies that is done. This one, you take a sample from the tumor, and this one is an invasive method. It's done early during diagnosis. But the problem with the traditional way of doing biopsies, it's that it's very difficult to repeat, and sometimes they have some risks. So what we want to be able to do is to just draw a sample from the blood and be able now to detect the tumor. So if we develop liquid biopsies, our goal and hope is that we're going to be able to now identify neuroendocrine tumors very early. I know that some of the patients, they have symptoms for many years, and they don't, and then the diagnosis happens really late during the disease. Hopefully this biopsy now will allow us for early diagnosis, but also we want to use these liquid biopsies to be able to monitor the disease progression, but also to monitor the response to therapy. And as you see here, this is a big project. It involves mainly the team of Dr. Simran Singh and Hong Leong at Sunnybrook, but also a lot of groups from Queen's University. So what are we looking for in the blood? So if you see here, it's a live cell. It can be a cancer cells, and cells, what they tend to do is to shed these what we call extracellular vesicles. So this kind of a bilayer is part of the cell, and this bilayer or vesicle, it contains different proteins or genetic material that really recapitulates what the cancer. So it's kind of a mirror image of the cancer. So if we go back to this analogy of a patient that would have type A and type B pancreatic neuroendocrine tumor, if we do a liquid biopsy, so in the blood, we should be able to find these circulating vesicles that are coming from the type A tumor and type B tumor. So this, first of all, it can tell us very early that the patient has a cancer, and then hopefully also will be able to tell us whether it's type A or type B. But again, going back to the thing I mentioned before, is to monitor the disease progression to see whether they respond to therapy and so on. So as I mentioned, in these vesicles, they have different types of genetic material. What we are interested for is something that we call microRNAs. You should think about microRNAs as the maestro of the orchestra. So these are very powerful, very small molecules that are very stable, but they have the ability to really orchestrate how a cancer cell will function. And we've done a lot of studies, and we understand now some of the role of microRNAs. We know they are very important, but they also now will be able to detect them in these extracellular vesicles that circulate in the blood, and again, they're coming from the cancer cells. So as a proof of principle, this is a study that was initiated by Dr. Renwick and Hong Leong. They took samples with no net diagnosis. These are our controls. We call them healthy volunteers, and also we took blood samples from patients that have a net diagnosis. And then we looked for the different levels of microRNA circulating in the blood. So here I'm showing you a graph. These are the levels of one of the microRNAs. I'm going to call it as microRNA net here because it's still at the stage of validation. But if you see here in the first column, what you see here, these are the levels of this microRNA in the blood from what we call healthy volunteers that they didn't have a net diagnosis. And as you see, most of these patients, they have 0 levels of this microRNA except one of them. But now if you see now the patients that they have lung neuroendocrine tumors, they have a very high level of this microRNA, and the same thing with patients with GI nets, compare obviously with the controls that have no. Again, here you can see that we have different levels. We don't know yet if that still has to do with how big the tumor is or whether there is metastasis, but this is something we want to understand. So this is one of the tests we're trying now to develop at Sunnybrook. The next step is to validate whether we can use this microRNA test first for diagnosis. So the group of Dr. Simran Singh and Hong Leong are now recruiting patients at Sunnybrook. So if somebody visits Sunnybrook in the future, will be asked to whether they can donate some plasma or some blood, where we can go back and validate this microRNA. And our hope is that we'll be able to take this in the clinic within the next, hopefully, few years. So, I mean, the other thing we're trying to do is also to enable our pathologists to better differentiate what we call type A and type B. So our pathologists now, they can do very well, like say if you have I think most people will be able to differentiate very well what's a green apple and a red apple. But if I ask you to differentiate between the red tissues and the empire, you might have some difficulties, right? So what we want to do is to help our pathologists to be able exactly to pinpoint whether patients have a type A or type B neuroendocrine tumors during the initial tissue biopsy. To do that, we took advantage of existing samples at Sunnybrook. So Sunnybrook and the Susan Leslie Clinic operates for more than 10 years. So the good thing is that they were always biome banking to a certain level tissue. So with a grant that we received from CNETS 2 years ago, we went back and we found all of these samples, and now we're building what we call tissue microarrays from around 800 patients. And this now it's a very, very powerful tool, because now we're going to go back and analyze these samples. And the idea is now we'll be able to identify markers that will be able to tell us, okay, this is a green ampoule, this is a red ampoule, and this is somewhere in intermediate, maybe it's a transition from the green to red, and this is a honey crisp, and maybe you need a different approach. So we're very close to finish building this tissue microarray, and we are very excited about what's coming after. So I'm going to switch now to talk a little bit about the models we're trying to build to find therapies, and what we call is organoids or PDX models. So I did mention that, you know, from the surgical specimen, if there's a surplus extra fresh tissue, then this will come in our group. And we do two things. One is to develop what we call patient-derived organoids. So this is like a miniature tumors, small tumors that they grow in these 384 well plates, which we use in the lab. And the second thing we use, it's what we call a patient-derived xenograft models, and I will show you an example of this. So first I will talk about these organoids. So what's the advantage of this? The advantage of this is that from a sample that we get now from a patient, we can make up to 1,000 of these small organoids when you see here in the individual wells. And now that gives us the capability to go back and screen for drugs. Let's say we have 100 drugs that are candidates to use for neuroendocrine tumors for a patient. We can take the sample from the patient, make these organoids, and screen, let's say, 100 FDA-approved drugs, and maybe that will enable us to identify new treatments. So here I'm just going to show you some beautiful images that we get in this collaboration with Dr. David Andrews. So here you see different types of organoids. The top one is from a primary tumor. The bottom one is from a metastasis from the same patient. And now what we can do is not just grow them, but use different markers to understand how they grow, how fast they grow, how they respond to therapy, whether they are undergoing stress, you see here with red, or whether they are dying as shown here with green. And not only we can do this for the organoid, but actually with this technique we can actually look at individual cells. What you see here is now if we take sections from the bottom organoid of the organoid and go in the middle, you can see the different cells and how they behave. And again, we can do this and with the different markers. So some of the initial results we had is, for example, if you take again, this from the same patient, you take a sample from the primary tumor, and this in case mesenteric metastasis, and you grow these organoids. The first thing you will see is that actually the organoids from the metastasis, they grow faster. This is something that we always suspected, that metastasis is a bit more aggressive, and now we can recapitulate that and see that in the lab. The second thing that we suspected we find and we found is that now if we use a treatment, I call this treatment A, in fact it's a treatment that is used in the clinic, and treat these organoids. So with blue, now you see these are the organoids from the primary tumor. If you look at the fourth column here, which says 5 micromolar, and compare it with the control, which is DMSO, you can see now that these cells are start dying. So the primary tumor in this case will respond to therapy. However, if you look at the same now patient, but this is the sample from liver metastasis, and you compare again the first and the fourth column, you can see there is no difference. So this tells us that most likely if this patient is treated with this treatment A, the primary tumor will respond, but the metastasis will not. And really our goal is to try to find ways to treat metastasis. And hopefully in the next few years we'll be able to find some of these new treatments. Now the second approach is we did what we make these avatars from the tumors. We call them patient-derived xenograft models. This is a collaboration with a group of Dr. Hong Leong. And hopefully this one will play. Let's see. I have a video here that I would like you to see. Let me see if I can get this going. Okay. Okay. Okay, good. I hope you are able to see this video. So basically, what you saw there is that we do get these embryos from these eggs, or is a duck eggs, which we cultivate in the lab. And the advantage of this one is that they are very vascularized, so it enables us to engraft pieces of the tumor that actually we get from the clinic, and that's a different way to do it. The advantage is that it preserves better the architecture of the initial tumor. So again, as I mentioned, is a collaboration with Dr. Hong Leong. So far, these again are numbers from September, but we were able to do derive these models from 6 patients. 5 they had small bowel net, one of them was colon. We've done it both from samples from primary lymph node metastasis and liver metastasis. And as you can see here, the tail grade is really high. It's around 80%. So I'm going to show you now some examples. That one of the things we do is to want to make sure that the histology or the appearance is preserved. So if we had a type A tumor, we want to make sure that when we put them in this what we call PDX ovals, they remain type A, and if they are type B, they will remain type B. So to orient you here, what you see on the outside is the chorial membrane. This is from the egg, and what is in the middle, this actually is the tumor that was engrafted from the patient. And to orient you a little bit more, what you see here with yellow marked with yellow, actually these are the tumor cells, neuroendocrine tumors. They form these what we call tumor nets or tumor neighborhoods, and what you see around is what we call stroma cells. So these are supporting cells for the tumors. And this from a patient with pancreatic neuroendocrine tumor. And if you look, this now is from a liver metastasis. The architecture is a little bit different, but again, here in the middle and outside here, again, you can see these tumor nets. So we start now characterizing this, and one of the markers we use is called chromogranning. This one stains the cancer cells, and again, you can see that these tumors now engrafted in this model, they do retain the characteristics. It's one of the main characteristics for neuroendocrine tumors. And now we're trying to see whether we can also see the different subtypes, type A or type B we talked about. So as we mentioned on the top now, you can see some examples. This is what we call a bright field imaging. So circle with red, these are the tumors. But what we can do, which is really exciting, is similar to what they will do in the clinic. We can use ultrasound, so we can see the tumor size, and then we can also see whether this tumor is vascularized, whether there is a blood flow inside the tumor. And that's very important because we want to start treatments now. We're going to be able to understand whether these tumors are actually respond to the different treatment types. And here you see the quantification of the volume and the vascularity of some of these. So what are the next steps when it comes to identifying new therapies? What we really want to do is first to start with the patient-derived organoids. That gives us the ability to screen for high number of FDA-approved drugs. And then once we identify the ones that are the most promising, we're going to go back and verify them using what we call the PDX oval. Obviously, the idea is that now at some point we'll be able to find therapy specific for the different type of neuroendocrine tumors. And that brings me to one of my last slides, which is what we try to establish here at Sunnybrook, what we call a net translational research program. On the Right side here, you see that we're trying to define new biomarkers, both tissue biomarkers, but also liquid biopsies. We want to understand how these new biomarkers associate with the traditional use that we use in Sunnybrook, like imaging, and hopefully we'll be able to incorporate these new types of biomarkers in the clinic. On the left side, as I've shown, now we're using the biospecimens we get from patients. We want to establish organoids, do drug screening, identify new drugs, validate them with a PDX-OVO model, and hopefully later on do clinical trials and move them back into the patients. So I would like to acknowledge, as you can see, it takes really, usually we say it takes a village to establish a translational research program. There are a lot of teams involved. What you see here on the left is my team, members of my team. I've shown Nilakshi Kulathunga, who is here, which is really heading this project, along with Zoe Wang, which is a PhD student in my group. Sunnybrook in the Susan Leslie Clinic, I've been very lucky to find a very enthusiastic response by all the clinicians, and I really would like to thank all of them for their support, especially Dr. Simon Singh, Jose Rodriguez, Calvin Lowe, Julie Hallett, and Wei and Juan and Lina Chen are our pathologists who help us with the developing of tissue biomarkers. The SRI biome banking, that was a very important step, and Dr. Hubert Tsui with his team helped us to set this up. And as you've seen, there are many scientists who are collaborating with both at Sunnybrook and at Queen's University. I also like to thank the funding agency, especially CNET. CNET was the first funding agency that funded one of my projects in my group, and that was really important for us to start. And then we got a grant from Neuroendocrine Tumor Research Foundation in the States to develop the different preclinical models, and recently we got another grant for CIHR that will allow us to understand the different subtypes. Last, but the most important thing for us, and what I would like really to thank, is all the patients and their family members. Without them, we wouldn't have been able to do any of this. And I do really hope that some of the work we do here one day will help both the diagnosis and the treatment. One thing more to mention is that we are welcoming patient advocates and patient visitors in my group. A lot of the people in my group, sometimes we work until midnight to derive these models, and I think it's very important for them to connect with patients and their families. But I also hope that patients and families will find it somewhat exciting to come in the lab and see what's going on and meet us. So I will stop here, and I'm very happy to take any questions. Thank you so much.
Okay, I'm unmuted now, and I guess I should put my webcam back on. Okay. Here we go. Yeah, well, thank you very much. That was fascinating and very complex, and I'm sure that everybody's got more questions than answers to all this. So I encourage everybody who could put their questions into the chat function. Just post in your questions, and we'll take some as we go along. I'll see what's there now. Okay, the first question we have for you is, if we're a patient at Princess Margaret Hospital, can we still participate in clinical trials at Sunnybrook?
So I want to differentiate what we're doing is not clinical trials per se, so we're not testing new drugs, but if you're undergoing a surgery, I mean, we have to have an MTA agreement with Princess Margaret. but we are able to take tissue, and is Pieces-Mascaret not far? So the question is, it's yes, we should be able to get tissue and bring it here in the lab. And the same thing goes for plasma. For plasma, it might be easier if somebody comes here at Sunnybrook and donates plasma or blood, sorry. But yeah, please contact me. I think it should be feasible, especially for blood. It's going to be much easier.
Okay, the next question is a bit similar. Are patients local to Toronto but not treated at Sunnybrook able to participate in the biobank program? You've kind of addressed that, but can they be self-referred or referred to another oncologist?
Yeah, I never thought about that. Really, thank you for putting this up. I think because, okay, if your primary site is not Sunnybrook, obviously it's not somebody from the clinicians here. But if you want to participate to the biobank, I think the best way now it will be to contact me, and then I will see how we can coordinate this with our biobank. So the patient will have to come in and donate blood at some point, which is that's feasible. So right. I don't think any need for a referral for the biobanking.
Okay, so we know who to come to. Liquid biopsies are only, quote, good if doctors ask for it. Are doctors aware enough about NET to request liquid biopsy before it's too late?
Yeah, I mean, yeah, if we have a liquid biopsy and the primary doctor, I would think the family doctor is not aware they exist, and the patient will have symptoms. But our hope is that at some point, once we establish these liquid biopsies, then patients that have some symptoms and there is no diagnosis, will refer them and to get these liquid biopsies. So hopefully it will be like a mammogram that is done for breast cancer one day, or like the PSA that is done for men for prostate cancer. We are not there yet, but that's our vision. That will make it so easy that it shouldn't be a problem for somebody to get it.
Just to add on to that, how many years away is that?
So the first one, the one I show, it's we hope to validate it this year. So it's really a matter of numbers. So the more people will be able to enroll, and it will make stronger the evaluation. I think that will be easy. And then we have to move it to become what is called a certified test. And I think that will take a few more years, but it's not. So I would say within hopefully the next 3 to 5 years, we hope to be able to offer it. That's my hope, but let's see. We also, I mean, the good thing is that we already tried to test this in ongoing clinical trials. So we're kind of already getting samples from patients that are in different clinical trials. And you know, because there is not too many tests out there now, a lot of the physicians, not only in Canada, but also in US and in Australia, we have collaborators in Australia that are participating in this. So it seems there is a lot of excitement. Hopefully we'll be able to bring this into the clinic. That for me would be my #1 goal right now.
Okay. Can liquid biopsy replace urine samples to gauge how much excess hormones are being produced by functional NET?
Yeah, we're going to have to do the comparison for that, and I can't. That's again, it's a great question. It's something we're going to have to test. For that, we're probably going to have to do another. Once we verify that this biopsy it precisely can give us identify a patient with neuroendocrine tumor. The second is whether we can use it for disease progression, and I think at that point we like to ask patients to also give us urine sample at the same time, or biobank urine samples? Actually, I'm going to take a note because it's a very good question, and I'm wondering whether we can already start biobanking this from the same patients. Thank you. That's really good.
Yeah, it's a big problem. Does the chromogranin analysis picture shown give the same results as the chromogranin A blood test result?
I'm not going to go into, I haven't tested it. I know that the chromogranin A blood result, it has some problems, but I'm not the expert to talk about this right now. And of course, one of the things that we want to do is to compare the liquid biopsy that we're developing with the chromogranin A test that is done for the blood. And we hope that our test will be more sensitive and more specific. So we'll see.
Down the road. Okay. Oh, and then how do we contact you? I think at the first slide you had some information.
Yeah, I mean, Barbara, if you want, you can send my my email. I'm happy to give my email to everyone. But also, if you look at my name, if you put Iacovos Michael Sunnybrook, you will find my email, and please email me. Please don't call. But feel free to email me. I try to get back within 48 hours usually.
That's pretty good. If I'm a patient of Dr. Singh's, what do I need to do?
So I think if you're a patient, if you undergo surgery from now on, the past 6 months, definitely going to approach you to ask you to consent for the surgical sample. For the blood sample, hopefully starting February 1st, we'll start that. But also you can ask Dr. Singh that you can let him know that you're know about these liquid biopsies and you would like to participate, and we'll be able to arrange this.
Okay. With regard to the slide showing primary versus metastatic NET, is there any role of molecular imaging in identifying the primary site of the NET, if the NET is of unknown origin?
If the NET is not, sorry, so if you have, let's say, the type A that doesn't metastasis, whether we should do imaging, is this the question?
I can't, the primary versus the metastatic NET. So is there a role for molecular imaging in identifying, and I guess that's what we're talking about, the primary site of the NET, when I guess somebody's not able to find the primary?
Yeah, I'm not sure if I understand the question.
That'd be helpful.
But again, these are things, obviously, imaging and the practice that it's going right now, like the PET imaging, all this, they're not going to go away. The idea is when we do subtyping, it's to dive in more into the characteristics and be able to understand what type of tumor you have, right? So it's going to be at the level of either tissue biopsy or liquid biopsy. So we're not eliminating the imaging that is done right now. I hope that was the answer.
Whoever asked that question, if you want to put in another question to clarify your question, that would be helpful too. A huge thank you to Dr. Michael and their village, supporting them for focusing on NET tumors. Getting this diagnosis was difficult, especially when minimal treatments exist for NET. So thank you to you and your staff. So there you go.
Thanks so much. I mean, one of the challenges I knew when we started this was that these tumors are rare, and but that was also what we want to tackle, because there are no therapies, not too many treatment options right now. But the fact that we the volume is very high here at Sunnybrook. It's really encouraging, and we hope that we'll be able to make a difference. Thank you so much.
Right, yeah. When you speak of the therapies that you're testing, are you talking about chemotherapy type things or radiation? I mean, or a whole gamut? I wasn't clear on what you meant by the therapies that you're testing.
Yeah, so these are most of them. So what we want to do first is use FDA-approved drugs, and these are small molecule therapies, kind of be a chemotherapy type, or also small chemicals that they inhibit different proteins. So it can be like a tyrosine kinase inhibitors, if we want to talk more scientifically. And the idea is we want to do this because if the drug is already FDA approved, it's much easier to move it into phase 1 or phase 2 clinical trials, and hopefully bring it faster to the clinic. And specifically, we intentionally go with FDA approved drugs. So these are single therapies, but what we also do is a combination therapy with the to enhance the efficacy of PRT, or peptide receptor radionuclide therapy, which a lot of patients probably are familiar with. This it's a type of radiation that is used for patients that have metastasis and is not resectable. What we see is that maybe 20% of the patients we have regression, 50% of them are stable disease. What we would like to find is drugs that we can combine with PRT, so we have increased the number of regressions, so we see better response in the clinic. And again, we're using FDA-approved drugs, so the idea is that we'll be able to move faster into real clinical trials with patients.
Okay, and just one last question again from me. You know, the topic was net research projects at Sunnybrook. You have a wonderful, great one. Are there other projects going on at Sunnybrook that are related to nets, or are you sort of the umbrella group to look after that?
So these are the research projects, but there are a lot of clinical trials that are all going at Sunnybrook. Of course. And I'm happy to share the link. They're already at Sunnybrook. There is a website where you can look at clinical trials specifically for neuroendocrine tumors. Barbara, I can send you the link, and you can share it if you want. But maybe in the future you can have Dr. Simran Singh presenting. He's leading all of these clinical trials, and it's really exciting because there are so many. And our Sunnybrook, it's really one of the leading centers in the world when it comes to clinical trials.
Yeah, that's great. Well, on behalf of CNETS, I really appreciate this presentation and you giving your time to share your research with us. It's great to have these sorts of things going on. Thank you.
Thank you so much, and I would also like to thank CNETS one more time for the funding and the support. It's been very important for our group, and one more time the patients and their families. And we hope that we'll be able to make a difference. And again, if anybody wants to connect with us, come and see what we do here at Sunnybrook, feel free to get in touch with me. Thank you so much.
Okay, thank you.
Great, thank you. Bye-bye.
Okay, bye-bye.